By 1978, I could write, compared to almost any other object that starts with the letter D, DNA is very safe indeed. Far better to worry about daggers, dynamite, dogs, dieldrin, dioxin, or drunken drivers than to draw up Rube Goldberg schemes on how our laboratory-made DNA will lead to the extinction of the human race. Later that year in Washington, D.C., the Recombinant DNA Advisory Committee, RAC, of the NIH proposed much less restrictive guidelines that would permit most recombinant work, including tumor virus DNA research, to go forward. And in 1979, Joseph Califano, Secretary of Health, Education, and Welfare, approved the changes, thus ending a period of pointless stagnation for mammalian cancer research. In practical terms, the outcome of the Asilomar Consensus was ultimately nothing more than 5 sad years of delay in important research, and 5 frustrating years of disruption in the careers of many young scientists. As the 1970s ended, the issues raised by Cohen and Boyer's original experiments turned gradually into non-issues. We had been forced to take an unprofitable detour, but at least it showed that molecular scientists wanted to be socially responsible. Molecular biology during the second half of the 1970s, however, was not completely derailed by politics. These years did, in fact, see a number of important advances, most of them building upon the still controversial Boyer-Cohen molecular cloning technology. The most significant breakthrough was the invention of methods for reading the sequence of DNA. Sequencing depends on having a large quantity of the particular stretch of DNA that you are interested in. So it was not feasible, except in the case of small viral DNA, until cloning technologies had been developed. As we have seen, cloning, in essence, involves inserting the desired piece of DNA into a plasmid, which is then itself inserted into a bacterium. The bacteria, allowed to divide and grow, will then produce a vast number of copies of the DNA fragment. Once harvested from the bacteria, this large quantity of the DNA fragment is then ripe for sequencing. The 2 sequencing techniques were developed simultaneously, one by Wally Gilbert in Cambridge, Massachusetts, Harvard, and the other by Fred Sanger in Cambridge, England. Gilbert's interest in sequencing DNA stemmed from his having isolated the repressor protein in the E. coli beta-galactosidase gene regulation system. As we have seen, he had shown that the repressor binds to the DNA close to the gene, preventing its transcription into RNA chains. Now he wanted to know the sequence of that DNA region. A fortuitous meeting with the brilliant Soviet chemist Andrei Mirzabekov suggested to Gilbert a way, using certain potent combinations of chemicals, to break DNA chains at just the desired base-specific sites. As a high school senior in Washington, D.C., Gilbert used to cut class to read up on physics at the Library of Congress. He was then pursuing the holy grail of all high school science prodigies, a prize in the Westinghouse Talent Search. He duly won his prize in 1949. Years later, in 1980, he would receive a call from the Swedish Academy in Stockholm, adding to the statistical evidence that winning the Westinghouse is one of the best predictors of a future Nobel. Gilbert stuck with physics as an undergraduate and graduate student, and a year after I arrived at Harvard in 1956, he joined the physics faculty. But once I got him interested in my lab's work on RNA, he abandoned his field for mine. Thoughtful and unrelenting, Gilbert has ever since been at the forefront of molecular biology. Of the 2 sequencing methods, however, it is Sanger's that has better withstood the test of time. Some of the DNA-breaking chemicals required by Gilbert's are difficult to work with, Given half a chance, they will start breaking up the researcher's own DNA. Sanger's method, on the other hand, uses the same enzymes that copies DNA naturally in cells, DNA polymerase. His trick involves making the copy out of base pairs that have been slightly altered. Instead of using only the normal deoxybases, A's, T's, G's, and C's, found naturally in DNA, deoxyribonucleic acid, Sanger also added some so-called D-deoxybases. D-deoxybases have a peculiar property. DNA polymerase will happily incorporate them into the growing DNA chain, i.e., the copy being assembled as the complement of the template strand. But it cannot then add any further bases to the chain. In other words, the duplicate chain cannot be extended beyond a D-deoxy base. Imagine a template strand whose sequence is GGCCTA, GTA. There are many, many copies of that strand in the experiment. Now imagine that the strand is being copied using DNA polymerase in the presence of a mixture of normal ATG and C, plus some D-deoxy-A. The enzyme will copy along, adding first a C to correspond to the initial G, then another C, then a G, and another G. But when the enzyme reaches the first T, there are two possibilities. Either it can add a normal A to the growing chain, or it can add a D-deoxy-A. If it picks up a D-deoxy-A, then the strand can grow no further, and the result is a short chain that ends in a D-deoxy-A. DDA:(CCGGDDA). If it happens to add a normal A, however, then DNA polymerase can continue adding bases, T, C, etc. The next chance for a D-deoxy stop of this kind will not come until the enzyme reaches the next T. Here again, it may add either a normal A or a DDA. If it adds a DDA, the result is another truncated chain, though a slightly longer one. This chain has a sequence of CCGGATC-DDA. And so it goes every time the enzyme encounters a T, i.e., has occasion to add an A to the chain. If by chance it selects a normal A, the chain continues. But in the case of a DDA, the chain terminates there. Where does this leave us? At the end of this experiment, we have a whole slew of chains of varying lengths copied from the template DNA. What do they all have in common? They all end with a DDA. Now imagine the same process carried out for each of the other three bases. In the case of T, for instance, we use a mix of normal ATG and C plus DDT. The resultant molecules will be either CCGGA DDT or CCGGATCA